Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in this video I will be sharing the last part of photosynthesis. I think I said in the previous video that photosynthesis is a short section. It's a very short chapter. Usually it takes about a week or 10 days, depending on how many hours of class you have to get through it. So it's not that long, and it's also not as complicated as students tend to think it is. But if you have any questions, especially about the Calvin cycle, please by all means post them in the comments and I promise to get back to you. I've also seen lots of requests for students asking me about past questions and saying, please, can you solve some past question papers? So in the video, I think after this one, I will be posting 2018 papers, like questions from 2018, paper 1 and 2. That's for AS content, just so you can see how some of the questions should be answered. But before then, let's get into the last part of photosynthesis, where I will be discussing the factors that affect photosynthesis, C4 plants, and how light absorption works in photosynthesis. I think I will be starting with C4 plants before we go on to factors and then go on to light. So let's get started with C4 plants. So C4 plants are an aberration to the normal plants. So remember we said when we were discussing photosynthesis in the previous video that you have two stages of photosynthesis. You have the light dependent stage, which happens in the thylakoids. And what happens during that stage is simply light allows for photoactivation of electrons, and those electrons travel through electron transport chains, either in a cyclic manner or in a non-cyclic manner. And what that helps to do is provide energy for the coupling of ADP and phosphate, as well as protons for the reduction of NADP to NADPH. And those two things are then passed on to the light independent stage, which is called the Calvin cycle. And in the Calvin cycle, you have CO2 coming to react with RuBP, which is ribulose bisphosphate, and that forms a 6-carbon intermediate, which is then split into 2 molecules of a 3-carbon compound called PGA. And PGA is then reduced to glycerate phosphate using hydrogen from NADPH, and from glycerate phosphate we're able to make glucose, and the rest of the PGA, of the glycerate 3-phosphate rather, is used to regenerate RuBP. Now if that was confusing to you what I just said, please by all means go to the previous video and watch the last few minutes again. I think the last 7 minutes you would be able to get it. If you don't get it still, please post a question in the comments. Don't be shy. I always tell my bio students that there are no stupid questions. So even if you feel like this question seems totally unrelated, still ask it. If I know the answer, I'll tell you. If I don't, I hope someone from the community of watchers would be able to answer your question. But with all that said, with C4 plants, there's a little bit of a difference. Like we said, we have the Calvin cycle where we have these 6 carbon compounds. Now, when we speak about C4 plants, examples of such are maize and sorghum. They act differently when it gets to the light independent stage. And the reason for that is because there's a problem with rubisco, which is something I will explain to you just now. So, C4 plants like maize and sorghum don't carry out the light independent stage directly. They need to go through a series of navigating steps, or should I say detours, before they get to the light independent reactions happening correctly. And the reason for this is that the enzyme that catalyzes the reaction between CO2 and ribulose bisphosphate, which is called rubisco, has a problem. The problem with rubisco is that it catalyzes the reaction of oxygen with RuBP as well. In other words, rubisco likes to catalyze the reaction between CO2 and RuBP, and it also likes to catalyze the reaction between O2 and RuBP. Now, when rubisco catalyzes the reaction with oxygen, that is called photorespiration. And the problem with photorespiration, I'm just going to underline it here so you can see that I've put quite a bit of words on this slide, because I feel like students often find it a lot better when they're able to read whatever it is that's being said, or if they just want to take extra notes for later. So it's called photorespiration, and when you have photorespiration, it simply means that rubisco is catalyzing the oxidation of RuBP, which means RuBP would not be able to go through the Calvin cycle as it normally would. Because think about it, if you add oxygen to RuBP, which is a 5-carbon compound, you will not have the formation of a 6-carbon intermediate that will then split into 3-carbon compounds and then carry on until you have the formation of glucose and regeneration of RuBP, which then means that the RuBP is being wasted because it doesn't make the compound that we need it to make. So this process of photorespiration means that we have less photosynthesis happening. Photorespiration is also favored when temperatures are high and when the light intensity is high, and those are the places you would typically find plants like maize growing. And so plants like maize have sort of evolved some form of adaptation in order to allow them avoid photorespiration, and I'm going to explain that adaptation to you in detail. So how do C4 plants prevent photorespiration? What they do is that they prevent rubisco and RuBP from coming in contact with oxygen. Because remember what I said on the previous slide, that whenever rubisco sees oxygen and RuBP together, it just imagines them as a good match, but that is wastage for the plant. So we don't want that to happen. So C4 plants typically don't want that to happen because they would not be able to make any of the glucose that they are supposed to make and store in their leaves or in their roots and so on. So what they do is that they arrange rubisco and RuBP in what we call the bundle sheath cells. And I hope you've been able to see these cells in chapter 7 when you did things like plan drawings in the classroom. They arrange them in bundle sheath cells, and by doing this, rubisco and RuBP don't have any contacts with air in the leaves, so they're isolated. What then happens is that the C4 plants would then absorb CO2. When they absorb CO2, instead of the direct light-independent reaction or the direct Calvin cycle reaction where CO2 simply combines directly with RuBP, they will take that CO2 and they will combine it with a compound called phosphoenol pyruvate, also referred to as PEP. They combine that 3-carbon compound with CO2, and that makes a 4-carbon compound called oxaloacetate. If you remember your respiration well, you will remember that we came in contact with oxaloacetate. It's a 4-carbon compound. And the process of doing this is that they're simply isolating CO2 from the air. Remember that air is a mixture of gases. They're isolating CO2 from the air, they're combining it with phosphoenol pyruvate. So phosphoenol pyruvate is sort of like a carrier of CO2. It becomes oxaloacetate, which is a 4-carbon compound, and I'll tell you what goes on the next slide. And now, once that oxaloacetate is formed, it is converted to malate. Malate is also still a 4-carbon compound, if I remember correctly. And malate is the one that is then able to travel to the bundle sheath cells where rubisco and RuBP are being hidden. When malate gets into the bundle sheath cells, it is decarboxylated, which means CO2 is removed from malate and then added to RuBP, and rubisco is allowed to catalyze the reaction. And from there, the Calvin cycle can then continue. What this simply means is that C4 plants have to use a sort of meandering path in order to get to the light-independent reaction, because they can't afford to just have their CO2, RuBP, rubisco exposed in places where there is high oxygen concentration. If the oxygen concentration is high, then rubisco would typically add oxygen to RuBP and waste RuBP. So what they do is they take CO2 from the air, they combine it with phosphoenol pyruvate to make oxaloacetate. Oxaloacetate is converted to malate. Malate travels into the bundle sheath cells, and when it gets there, CO2 is removed from it, and that CO2 is then combined with RuBP, and the light-independent reactions continue from there. Something that's very important to also note about C4 plants is that they grow in very hot climates, and I think I've said this before. So you need to bear in mind that when you're dealing with a C4 plant, the enzymes of that plant have a higher optimum temperature. For example, PEP carboxylase, which is the enzyme that allows for the formation of phosphoenol pyruvate being added to CO2 to form oxaloacetate, functions at about 45 degrees Celsius, and the same enzyme in a C3 plant, a C3 plant being a plant that just allows the direct combination of CO2 with RuBP, only functions at 30 degrees. So the optimum temperature for enzymes in C4 plants are really high. So I hope that you understood that. And again, always know that if it just seemed a bit confusing, you can always ask a question in the comments. With that said, we can then just have a quick look at the factors that affect the rate of photosynthesis. Given that we said photosynthesis has things to do, like it relies on CO2, for example, if there's no CO2 present, then photosynthesis can't be completed because you would only have the light dependent stage of the process, but the light independent stage, which is the Calvin cycle, would not be able to happen. That then goes without saying that CO2 is a factor that would affect photosynthesis. Temperature is also important because, just like I said on the last slide, that the enzymes that help with the Calvin cycle, in particular in the stroma, need an optimum temperature at which they must function. So temperature is definitely important. Light intensity as well, and I will tell you why this is important in slides that follow. But when you have light hitting the photosystem, certain intensities of light will encourage photosynthesis, and the wavelength of light is also very, very important. So if you look here at these graphs here, and I know that they're not perfect, these I got them like from very old drawings of the different things that affect photosynthesis, but I'm just trying to fill in the gap here because it's just, yeah, that looks a bit better, I hope. So if you look here, this is the graph of light intensity. And what this simply says is that when you're comparing the rate of photosynthesis at increase in light intensity, it gets to a point as you increase light intensity where the reaction is quick. And after a while, it starts to level off because increasing the light intensity further doesn't necessarily result in an increase in the rate of reaction. The reaction will proceed at what we would call its maximum rate. So once it hits its maximum, increase in light intensity more would not result in anything. In the same way, you then have the rate of photosynthesis versus increase in carbon dioxide concentration. And again, carbon dioxide has to be used systematically in the light independent stage. So after a while, I mean, when you start increasing initially, I'm just going to use a red thing here so you can differentiate. When you increase initially, you can see a quick increase in the rate of reaction as you increase CO2 concentration. But it gets to a certain point, and I think this is what we will call the turning point over here between these two. Gets to a certain point where that starts to level off because CO2 again has to be used in a series of steps, and there's no way to speed up the reaction. The enzymes that are involved in this, especially rubisco, has to couple CO2 with RuBP, and then at some point the rubisco is just saturated, right? And so the reaction will start to slow down. With temperature, always think of temperature in terms of what you would think if you're dealing with an enzyme. That's in regards to photosynthesis. So as you increase temperature, obviously the rate of photosynthesis will increase up until an optimum point, which is usually a range between here and there. And once you reach that optimum point, the reaction is at its peak. And if you increase beyond that, the enzymes that are involved in photosynthesis will probably start to denature because the temperature is too high, and that then results in a reduction in the rate of photosynthesis. So those are the explanations for these graphs. Now, in photosynthesis, we discuss something called a limiting factor. And when we discuss limiting factors, I usually try to get students to envisage it. So let's say, for example, we have a series of reactions. So I'm just going to write out a reaction here. We have A plus B will give us C. All right. C plus D will give us E. Okay. And E plus F will give us G. Don't worry, I'm not going to do this all the way up to Z, so don't panic. I just wanted to have 3 different reactions. So let's say this is reaction 1, this is reaction 2, and this is reaction 3, and this here is the final product P. All right, so what we're trying to make in this reaction is G. All right, but before we can make G, we have to go through all of these series of steps. When you have a series of steps like this, what would affect the rate of reaction is the reaction that is slowest. So for example, if I add A plus B and say, let's say normally A plus B takes 5 minutes, and let's say C plus D takes 2 minutes, and E plus F takes, let's say, 3 minutes. So that's a total of 10 minutes in order for us to make G. Now we come over here, I'm just going to write minutes so that you don't confuse them with the reaction numbers at the bottom. So here we go, minutes, minutes, and minutes. Okay, so Now let's look at this. Say something goes wrong and we end up spending 7 minutes here. Okay, so we end up spending 7 minutes on A plus B. And the product from A plus B is C. All right, it needs to just make that circle a little bit better. The product from A plus B, I'm going to use a black marker here, is C. All right, now if we spend 7 minutes making C, it means that we are slowing down the formation of C plus D, because C plus D is supposed to happen after 5 minutes. All right, so we've slowed this reaction down, and by virtue of slowing this one down, we've also slowed down E plus F, because C plus D will make E in order for E to combine with F and make G. So the slowest reaction in a series of reactions would affect the rate of an entire process. In the same way, when you're thinking about photosynthesis, the factor that is available in the lowest amount will affect the rate of reaction. So let's think about it this way. Let's say, for example, we have a reaction of photosynthesis and we have different concentrations of CO2. So let's say in reaction 1, we have CO2. I'm just going to write CO2 here and then I'll write different numbers. So let's say we have a concentration of maybe I'm just going to use moles, right? So let's say we have 3 moles in the first process, we have 5 moles in the other one, and then we have 15 moles in the next one. Now, when you have 15 moles, the reaction is going to be a lot faster because there's just a lot more CO2 available. So the process would occur like really quickly. Where you have 5 moles, the process might not be as fast as 15, but well, it would be okay. Relative to 3, where once the CO2 is exhausted, the reaction typically stalls or it stops. It would stall if there is hope for more CO2 to come into the reaction, but if there isn't, then it just stops. So it means that the low availability of CO2 stops the reaction or stalls it or slows it down in some way. So this is what we call the limiting factor, meaning that the factor that has the lowest amount available in any process or in the process of photosynthesis would result in the slowest rate of photosynthesis. So if you look at this graph here, I'm just going to erase these ones I put here at the bottom so that you're able to see. So if you look at this graph here that I've put on the slide, here's the rate of photosynthesis and we have light intensity. All right, so light intensity is increasing, and we know that as light intensity increases, at some point the reaction will level off. But now we're not dealing with just one factor influencing the rate of photosynthesis. In actual photosynthesis, a series of factors would affect the process continuously. So let's say we're now looking at temperature as well as CO2 in addition to light intensity. Now, if you look here, when there's high CO2 concentration and high temperature, the reaction proceeds for much longer before it levels off. Like, first of all, it's a lot faster, and then proceeds for much longer before it levels off. But if you look at where CO2 is high but temperature is not that great, it means that we have an abundance of CO2, but the temperature is not exactly perfect for the enzymes. So the enzymes work a bit slower than they typically would. And then you start to see that the reaction doesn't get to this peak over here. It levels off a lot faster. And the same thing then happens when you have low CO2 and low temperature. So when there's low CO2, it's like getting a person out of bed for a meeting that could have been an email, right? So the enzymes are like, why exactly are we here? So they're making whatever needs to be made. They're carrying out photosynthesis, but at a very slow rate because the CO2 production is really low. And the temperature is not great for the enzymes either. So that makes the rate of photosynthesis a lot slower and also levels it off really quickly. So this is how limiting factors influence the rate of photosynthesis. If I find a question on this, because I know that some of these things are hard to visualize when they're being explained, but I will try as much as possible to get a question that has this sort of problem and try to explain it in a video to you guys so that you're able to see what I mean. Now, when we understand limiting factors in photosynthesis, they can help us grow crops in controlled environments. So you've probably heard of a greenhouse, which is typically a house and it's often made of glass or transparent material where you can grow different plants under controlled conditions. So you can sort of figure out the amount of light that they need. You can figure out the amount of CO2 that they need, the amount of water that they need. You can sort of filter out the light intensity and all of those things. And for that, that reason, you're able to then grow certain plants that typically would not grow in outside environments where they are victims of natural forces, if I can put it that way. And this was an interesting thing for one of my students who did a research project with me, just understanding how greenhouses work and then trying to say, well, if that's the case, can we grow foods that are not in season if we're able to control the factors and give them the factors that they need all year round? Perhaps we'll be able to grow some of the staples that we typically can't grow out of season and, you know, not have to face world hunger. So I thought it was a very interesting proposal, and he did some experiments that yielded very good results. Okay, so that's it on factors. Now I want to speak about light. So the factors that we mentioned when we looked at the graphs, we spoke about light intensity, we spoke about temperature, we spoke about CO2 concentration. But something else that's also important that we mentioned, and I didn't have a graph for that, but here it is how the wavelength of light affects photosynthesis. Now, light is present in different wavelengths, and we have what we call the visible spectrum of light. So that's the light that we can see. And in that visible spectrum, there are different colors of light. These different colors are very important because they happen to occur at different wavelengths. But in addition to that, these wavelengths also influence which molecule of the chlorophyll or the chloroplast is absorbing light. So if you remember again, in the thylakoids, we said that we have this funnel arrangement. I'm just going to try to draw something over here. We have this funnel arrangement, and at the bottom of the funnel, we have what we call the primary pigment. And lining the funnel, we have accessory pigments that trap light and pass that light down to the primary pigment. All right. Now, the light that these accessory pigments are able to trap is very dependent on what wavelength of light is made available to them. So if you look here, this is what we call an absorbance spectrum. So if you can see that it's the absorbance of light. If you look here, there's a blue line and there's also a red line. The blue line shows us what wavelength chlorophyll A absorbs light at. So I think maybe I should use a green marker here so you're able to differentiate from the actual graph. So the blue line tells us what chlorophyll A absorbs at, and if you look at it, between 400 and I think this is about 450, chlorophyll A absorbs a lot of light, right? That's the absorbance spectrum. And between 450 all the way to 600, I mean, we have a tiny little peak here, but all the way to 600, we don't have much happening. So it means it doesn't absorb light between these two wavelengths. And then between 600 and 700, it goes on to absorb more light. So that's chlorophyll A. Chlorophyll B, on the other hand, also starts to absorb somewhere between 420, I believe, and it absorbs all the way to 500. And between 500 and 600, you can see it also doesn't absorb much, so let's say about 570 or 590. And then it absorbs between there to about 680. All right, so that just tells you the two different types of chlorophyll and how they absorb light. This is called the absorbance spectrum. We call the spectrum the absorbance graph or the absorbance spectrum, and this spectrum is usually called the red, blue, violet region. Carotenoids also absorb in the blue-violet region, so they absorb also somewhere in the blue area, while chlorophyll A and B absorb strictly within these wavelengths. And I'll tell you why this is important just now. So now, photosynthesis, we said, is dependent on the wavelength of light that's available. So if you take that graph we just looked at, which is the absorbance spectrum, and you try to compare it to what we call the action spectrum, you will see a great correlation there. So over here, you can see that between 400 and about 450-ish, maybe somewhere over there, I think here I can maybe use a red line. Between 400 and 450-ish, you seem to have like a high rate of photosynthesis, okay? Remember that both chlorophyll A and chlorophyll B absorb highly in this region. And between 500 and 600, they don't absorb that well. Both of them, like not between 500, but between 500 and about 580, they don't absorb that well. And between 600 down to 700, then you see a greater level of absorption. What this is telling us is that photosynthesis would occur really highly within the regions where the absorption of light is highest. So the wavelengths where light absorption is high would typically correlate with the regions where, or with the periods where photosynthesis is also high. So if you supply a plant with wavelengths between 400 and 450, for example, photosynthesis of such a plant would be really high because it's at the wavelength where the pigments are able to act, and they're able to absorb enough light. So they're able to carry out the light dependent stage of photosynthesis. If you supply light between, say, 450 to maybe 580, you can see that there's even a dip. So you might not have a great increase or a great rate of photosynthesis. This over here is called the photosynthetic action spectrum, and it typically just tells us that at certain wavelengths of light there is a high rate of photosynthesis because the pigments are able to absorb light at that wavelength. If this has been confusing, please ask me in the chat and I promise to explain it to you, but it is just really, really easy if you think of it that way. This brings us to the end of photosynthesis. It's a very short chapter, like I said, and I hope that you have found the videos helpful. I will be posting past papers next, so that on AS content, and I'll try to do that intermittently so that you're just able to see how to solve some of the difficult past papers that have been done by some of your predecessors, and I hope that you do better as you go along with these questions and solving them. So until then, have a good time. I will speak to you again soon. Bye.